طيب اسمعوا هالشيء العظيم هذا اللي صار. هذا الاسبوع الماضي خبر هيك طلع انهم الساينتست قدروا انهم يدخلوا نانو بارتيكلز على السيلز، وهاي النانو بارتيكلز بتشتغل بالريموت كنترول. اوكي؟ ف بالريموت كنترول they were able انه يشغلوا النانو بارتيكلز وتطلع ليزر. اوكي؟ So just imagine the consequences, implications. يعني انت تخيل ممكن تدخل drug specifically على cancer cell. Once you're sure انه دخل على cancer cells كبسة زر تك اشتغل الدراج قتل الكانسر cells. Really nice, huh? It's amazing. I don't know كيف بفكروا. I don't think انهم بفكروا ابني رايح ندرس بهذا ولا لا، راح يكسر الاضراب ولا لا، ادفع فاتورة الكهرباء كيف. السيارة لازم اصلحها، لازم اروح عند ميكانيكي اتهاوش معه. كل هاي الديلي لايف mundane issues. ايه اي فترة هاي؟ الاكتشاف بسمع الموقف. خبر بسمع الموقف طلع بسمع الموقف. بسم الله. نحكي عن شيء حلو. Metabolism in erythrocytes. So we'll be talking about 3 metabolic pathways or 3 metabolites or 3 reactions. The first is the 2,3-bisphosphoglycerate. The second is glycolysis, specifically pyruvate kinase. And then we'll talk about the glucose 6-phosphate dehydrogenase and the Pentose phosphate pathway and why it is important in erythrocytes. So let's start with this. So we talked about this before, and we said that 2,3-bisphosphoglycerate is a byproduct, يعني product secondary, ثانوي, فرعي, byproduct of the glycolytic pathway. So what happens is that you have glucose, glycerol, phosphate, etc. 1,3-bisphosphoglycerate. It continues all the way to pyruvate. Like in this here is a byproduct, 2,3-bisphosphoglycerate. Now it can be integrated. It can go back into the glycolytic pathway, and the consequence of that is that there would be no production of ATP. Okay, so you can have this, and you have a phosphatase that removes a phosphate, but it goes to waste. So it is a waste, sort of. But 2,3-bisphosphoglycerate is important. We talked about it before in the second lecture, regulation of hemoglobin. And we said that what it does is that it reduces the affinity of hemoglobin towards oxygen so that oxygen is released readily, quickly, easily, that is. So how does 2,3-bisphosphoglycerate bind to hemoglobin? Well, it sits right in the center of the molecule. And 2,3-bisphosphoglycerate has a number of negative charges, number of phosphates. And as a result, it can make interactions, several interactions with a number of amino acids. So in the core, in that center. So these interactions involve a lysine on both sides. You can have the amino group of both chains, and there is a histidine which is numbered histidine 143. So you can have this interaction. By having such interactions, you can have stabilization of the T structure of hemoglobin, which is the low affinity state of hemoglobin. And this happens in tissues. Great. So this histidine is important. And the whole interaction is important as well because the 2,3-bisphosphoglycerate is probably like the main driving force. It is, I'm not going to emphasize the role of this phosphoglycerate over protons and CO2 and all the other factors. But 2,3-bisphosphoglycerate is important because once it's removed, this is hemoglobin without 2,3-bisphosphoglycerate, really high affinity. This is normal level of bisphosphoglycerate. This is if you have high concentration of 2,3-bisphosphoglycerate. So you can have a shift to the right. And I got a few questions on what I mean by a shift to the right, because eventually you will get to the saturation level. But this is at very high concentration or pressure of oxygen. Okay? So when we say shift to the right or left, we're mainly focusing on P50. That is, what is the value of P50 right here? Okay, so that's what's important. So with the 2,3-bisphosphoglycerate, you have this shift to the shift of the curve to the right, lower affinity. Stabilization of the T structure, more electrostatic interactions. What's important about histidine 143 is that it is not present in fetal hemoglobin. It's not there. Now, it's not a mutation, it's just that it is a variant. Maybe in one of my lectures I said there's a mutation in fetal hemoglobin. I apologize. It's just a variant. Okay, it's not necessarily a mutation. So there is a variant. Our fetal hemoglobin does not have histidine 143, and as a result, there is reduced interaction between 2,3-bisphosphoglycerate and fetal hemoglobin. And as a result, you have this fetal hemoglobin having stronger or higher affinity towards oxygen. Okay. So that's the first one. Yes?
بالتالي السبب الوحيد إنه الفيتال هيموجلوبين الأفينيتي بتاعته أعلى بس بسبب.
No, there's another reason that I'll mention, I think, in this lecture. There's another reason. And the reason is that in the fetus, there is lower level of 2,3-bisphosphoglycerate. Okay. So let's talk about glycolysis in erythrocytes. Glycolysis is the main metabolic pathway in erythrocytes. There is no mitochondria in red blood cells, and as a result, the glycolysis is the only source of energy, the only source of ATP. Okay, so there's no Krebs cycle, there's no oxidative phosphorylation, electron transport chain, etc. So glycolysis is important, not only because it's a source of ATP, but it's also a source of NADH as well. And NADH is important for converting methemoglobin, which is the globin that has iron, or heme having iron in the ferric state, that is inactive, cannot bind to oxygen. So it has to be converted back, has to go back to the normal hemoglobin molecule, and this requires NADH. And glycolysis is the source of NADH. Now, ATP is important as well for phosphorylating proteins and sugars. So many of the cytoskeletal proteins need to be phosphorylated. Now, the red blood cell has to be sort of like flexible. It has to change its structures so that it squeezes in the narrow capillaries, right? And in order to be able to be flexible and to change shape, the cytoskeletal proteins must be phosphorylated as part of regulation. And ATP is needed for that. In addition, ATP is needed for membrane asymmetry, that is the differential distribution of proteins on the plasma membrane of red blood cells. It's important for the function of ion pumps. Ion pumps are important as well for regulating the amount of potassium and sodium inside the cells, and that is important as well for maintaining red blood cells, and not only the function but also the shape of these cells. Now, it's a category oxidation, NADH for reduction. Did I say? I'm sorry, the reduction of methemoglobin. Sorry. Okay, ATP is needed for, again, cytoskeletal proteins for maintaining their structure. And this is, again, the glycolytic pathway. Glucose, cleavage into two products, and all, you have pyruvate at the end. And then pyruvate is converted to lactate. and it goes back. And then gets released out. Okay, so let's talk about the very final reaction, which is phosphoenolpyruvate to pyruvate. And this reaction, you have a generation of ATP, and this reaction is catalyzed by pyruvate kinase. So pyruvate kinase is important. It's important because, well, yeah, it moves the pathway forward, but also it generates ATP, and there are different isozymes. You remember what isozymes are, huh? You know, it's the same, well, different enzymes catalyzing the same reaction, produced by different genes, different regulations, different kinetic properties, and so on. So there are different isozymes. You have an isozyme in liver, and you have an isozyme in muscles and in red blood cells. Now, there are differences in regulation, and that is the liver isoenzyme is inhibited by protein kinase A, but not the muscle enzyme. The liver enzyme is also inhibited by ATP, acetyl coenzyme A, alanine, long-chain fatty acids. It's all shown in here. So what you have in here is glucagon activates protein kinase A. It phosphorylates the enzyme. The enzyme is inactive when it gets phosphorylated. So the pathway stops. And at the same time, you have these factors regulating the enzyme, protein kinase A. Acetyl coenzyme A is an inhibitor, as well as ATP, long-chain fatty acids, alanine. This is an activated feed-forward regulation. Okay. The liver enzyme is also controlled at the level of synthesis. So if there is ingestion of carbohydrates, that stimulates a production of more proteins in the cells, and that is important. It's important لما نيجي نحكي عن a protein kinase deficiency. So we have the fetal pyruvate kinase. Now the isoenzyme in fetal cells, it has much greater activity than the adult isoenzyme, so the glycolytic pathway moves faster, and as a result, you have lower concentrations of the intermediates, including 1,3-bisphosphoglycerate and 2,3-bisphosphoglycerate. Okay, so if you have if this is moving really fast, it means that all of these intermediates, including 1,3-bisphosphoglycerate and the byproduct, the 2,3-bisphosphoglycerate, would be at lower concentrations, and as a result, less fetal hemoglobin binds to 2,3-bisphosphoglycerate, and it has this higher affinity. Okay, now what happens is that once the child is born, you have a switch from the fetal protein kinase A to the adult protein kinase A. So protein kinase A is an important enzyme. Now what's important about it is that it can be deficient. You can have certain mutations affecting this enzyme. And there are different types of mutations in pyruvate kinase. You can have mutations that can affect binding to the regulator, to the activator, the fructose 1,6-bisphosphate. So it's not activated as much. So you have slower reaction as a result of that. You can have mutations in the enzyme so that the Km would be affected, the Vmax would be affected as well, or the velocity would be affected. The enzyme is not as efficient as normal enzyme. Or you can have the stability that is altered. The enzyme gets degraded really fast. It's not produced. It's not as stable as normal. Now, the severity of the disease depends on how affected the enzyme is. Okay? Now, usually, normally in our normal cells, we do not need enzymes to be 100% active. If they are 50% active, it's fine. It's just fine. The person will be normal. But if it goes below like 30% or 20% normal level, well, no, that would be a problem. Of course, the more affected the enzyme is, the more severe the patient would be. Now, if there is deficiency or lower activity of protein kinase A, red blood cells will be affected because there is huge dependence on glycolysis, but not liver. The reason is that in the liver, well, it can compensate by other pathways in producing energy. That's one. Two, there's compensation in producing more of the protein kinase enzyme, because the way it's regulated is different. So you can have stimulation of production of protein kinase, of protein kinase in liver versus red blood cells. Because once you have this enzyme deficient in red blood cells, again, there's no DNA, there's no compensation in these cells. So this is deficiency of protein of pyruvate kinase. Sorry, I keep mixing protein kinase A with pyruvate kinase. Pyruvate kinase. Right? The third pathway is the glucose 6-phosphate dehydrogenase. This is the pentose phosphate pathway. And if you remember from your Biochem-2, when we talk about the pentose phosphate pathway, there are two phases. You have an oxidative phase and you have a non-oxidative phase. The non-oxidative phase is basically when you have 5 carbon sugars interconverting. So one changes to another, and so on. So producing different types of sugars. But in the oxidative phase, this is the series of reactions where you have conversion of glucose 6-carbon sugar to a 5-carbon sugar. Now this is an oxidative phase. Now you have oxidation of glucose. And this is catalyzed by a number of reactions. Now in these reactions, this is the reactions, and these reactions are irreversible. The very first reaction is the rate limiting reaction and the committed step. This is the slowest reaction. This is the one that determines that the pathway must move forward to completion. And it's catalyzed by glucose 6-phosphate dehydrogenase. Eventually, you have the كانوا مكتوب بالألماني اليوم الصبح هي غيرتها لأنه الصورة اللي أنا كنت حاططها كان عاملينها reactions reversible. It was wrong. It's one of I took it from one of the authentic biochemistry textbooks اللي بدرسوها بالعالم كله. مش عارف أي واحد فيهم. يا بناتي اسكتوا. الله يرضى عليكم. So I changed it مكتوب بالألماني. عموما, so eventually what you have here is the production of the glucose 6-phosphate gluconate, okay, with a carboxylic group. Okay, so. But notice what you have here is production of NADPH, machine, and you have production of 2 molecules of NADPH in these reactions, rather than NADH. And this NADPH is quite important. It is important for regenerating the glutathione molecule. Glutathione is an antioxidizing molecule. It's an antioxidant. It protects cells from the oxidizing agents, from the oxygen radicals, and so on. So regenerating the glutathione is really important, and it requires NADPH. And this is the only mechanism by which red blood cells can protect themselves from free radicals. That is glutathione. So you can imagine if you have deficiency of this enzyme, the glucose 6-phosphate dehydrogenase, this very first enzyme, it means that there will be no NADPH, there will be no mechanism, no way by which red blood cells can protect themselves from these molecules. And the membrane would be damaged. Molecules, proteins would be damaged as well. Okay, so that's the only mechanism by which red blood cells can protect themselves. Now, if there is no production of NADPH, it means that there will be low levels of glutathione, okay, and as a result, proteins will be damaged. Now, if proteins get damaged, and they get degraded, they would tend to cluster together. Once they cluster and aggregate, and red blood cells contain lots of huge amount of hemoglobin, if these cluster, remember that what they would do is that they would cluster and bind to the plasma membrane. Once they bind to the plasma membrane as clusters known as Heinz bodies, membranes are damaged. Okay, and the free radicals also damage membrane, and that leads to hemolytic anemia. Okay. So this is the importance of this glucose 6-phosphate dehydrogenase. Now, there is a condition known as the glucose 6-phosphate dehydrogenase deficiency. Okay, G6PD. Now, this is an important condition in the Middle East, especially in Jordan as well, because if you look at the distribution of the disease, you would notice that it really concentrates in the old world. Okay, so it is a major cause of disease, or its prevalence is high in Jordan relative to other countries. So you have to pay more attention to this condition. Now, the thing is that Africans, Mediterraneans, Oriental individuals are affected. The enzyme is located on the X chromosome, so males are more affected than females. Right. And it causes hemolytic anemia. Now, so what type of mutations exist in the G6PD gene? Mainly point mutations. You hardly have deletions or frame shift mutations. Now, that tells you something genetically. That tells you something that this enzyme is really important. It's critical for the life of a person. If you have major deletion or major mutation, like deletion or frame shift mutation, and if it doesn't exist, it means that this person dies early on. Okay? So all of these mutations that exist in this enzyme are basically point mutations. And there are different variants. There are different types, different mutations. And these mutations, there are different types. So you have the G6PD-B. So if we say variant B, it means that this enzyme is the normal enzyme. Now there are different types. We have, and the abnormal G6PDs are divided into different types or classes. So if you say class 1, class 2, class 3, etc. If we say class 1, these are the most severe. That is, the person will die in the young age. If we say class 4, this indicates that this deficiency is mild. It doesn't have major effect on the life or hardly any clinical symptoms for that person. Now, there is G6PD-A negative, and this is more common among Africans. فإذا قررتوا مثلا انكم تروحوا على أمريكا راح تشوفوا هذول البلاكس الأفريكان أمريكانز راح يكون عندهم this type of mutation more common than white people. Now, in these individuals, you have a point mutation again changing asparagine to aspartate that decreases enzyme stability. That is, the enzyme has the same activity, or it has some activity, normal activity, of course less because you have less enzyme. The enzyme is degraded. And then you have, so the disease is moderate because you still have some enzyme and you still have it active. Again, you don't have to have the enzyme to be 100% normal levels. All enzymes are active. You need some of it to be active. So, and then we have the G6PD Mediterranean. This is class 3. This is class 2. I'm sorry. This is a major condition. This is what we see here in Jordan, in the Mediterranean region. What you have in here is that the enzyme exists, except that its activity is almost negligible. It doesn't exist. Okay? So you have, again, different types. You have the G6PD-A minus. The enzyme is not stable, but the enzyme that exists, it does have some activity, which is enough to keep a person with mild clinical symptoms. But the G6PD Mediterranean, in this case, the activity is really affected. And it gets more serious with aging, with increasing age. The reason is that, for some reason, the G6PD enzyme, its level, is reduced as we age. Okay? So eventually this person would be really affected in the mid-age. Now, there is something important about the G6PD deficiency that you really have to know, is that it can really get worse under certain conditions. One, if certain drugs are taken by these individuals, such as the anti-malarial, antibiotics, antipyretics, what these cause is, again, because they cause oxidation. And as a result, and again, there is no NADPH, the red blood cells would be affected, hemolytic anemia, and so on. Fava beans, بالعربي بيسموها التفول. Okay, so if a person eats fava beans, the condition gets worse. The reason is that you have oxidizing agents and molecules in the fava beans, the food نفسه. Okay, so this can be serious in individuals as well as infection. And it makes sense. Infection, you have a lot of production of oxidizing agents during infections, and so on. But there is an advantage to malaria, because hemolytic anemia, again, okay, the parasite itself cannot complete its life cycle in these red blood cells because they die early on before they complete their life cycle, and so it can be sort of like an advantage. In places like Africa, where you have high level of malaria. Okay, so this is the lecture on the metabolism of erythrocytes. Any questions? مشيت سريع شوي. Questions? كأننا على رؤوسهم الطير. Okay. So let's just move on and start this topic, metabolism of iron. So I'll finish the first few slides, and then you can leave. So talking about, last week we talked about the biosynthesis of heme. Okay, so تفضل. بس حاطط حضور غياب by the way. كمان شوي. So شو صار؟ Okay, so we're talking about biosynthesis of heme. Few slides on the degradation of heme. So you have once you have death of red blood cells and release of the hemoglobin molecule that gets degraded and heme is released, iron must be preserved. It is an essential molecule. It has to be preserved. So we'll talk about it tomorrow, inshallah. So, but in terms of heme, it gets through a degradative process inside cells. And this is catalyzed by a number of enzymes, but eventually what you have here is the formation of biliverdin, and biliverdin is converted to bilirubin. And bilirubin is taken to liver, or this happens mainly in liver, and bilirubin is then conjugated, and it moves to the bile, from bile to the intestines. Okay, So in the intestines, you have a number of mechanisms whereby you have the conversion of bilirubin to urobilinogen. Some of it can leave the large intestine. There is a hepatic cycle, an intrahepatic cycle, and some of it, but most of it goes into the large intestines, where it can be converted to sterol-cobalin, and it gets out with feces. Now, some of the urobilinogen is taken in blood all the way to kidneys, where it can be converted to urobilin, and that gives urine the yellow color. Okay, so you have some urobilinogen in blood that goes back into the liver, and it goes back into the large intestines. Okay? So there are a number of conditions that are related to bilirubin metabolism, and we'll talk about it in the next lecture very briefly. هذا يعني بس هيك refresh your memories لأنه الموضوع مهم بالنسبة لكم بالنسبة للأطفال as well. Okay, so تاخذ حضور الغياب محلاتكم please.